Molecular Diagnostics, Introduction to the Molecular Diagnostics Laboratory. So when we think about how we diagnose disease, we've got various areas of the lab, and there's different things that they evaluate in those areas that will help us diagnose what's wrong with the patient. In clinical chemistry, we mostly evaluate analytes from blood samples to identify which cells or organs may be injured. In microbiology, we evaluate specimens for the presence of pathogens and attempt to isolate the pathogen to determine its identity and susceptibility to drug treatments. Immunology or serology, we use antibodies or evaluate the presence of antibodies to detect disease. Urinalysis, we evaluate the urine for abnormal constituents to determine the cause of disease or provide further evidence of disease. Hematology, we evaluate the blood cells to determine whether there are abnormalities that could cause disease. Cytology or histology, we evaluate cells or tissues samples for the presence of disease, namely cancer. In molecular diagnostics, we evaluate genetic material to identify the presence of select genes or sequences or errors within a gene. So we're looking at genetic material. So what is genetic material? Genetic material is DNA or RNA. And we're going to get into more of the details of what DNA is and what RNA is, and the various types of DNA and RNA that exist. But genetic material, in general, we're talking about something that has nucleic acids in it. Where does genetic material come from? Essentially, everywhere there are cells. So every type of cell, every fluid. There are a few cells that don't have nuclei. So red blood cells are anucleate, which means they don't have a nucleus. So there's not a lot of DNA inside of red blood cells. However, DNA and RNA can be deposited in the plasma from white blood cells or other cells. So DNA is still in the blood, even though big majority of the cells in the blood doesn't have any DNA. So molecular diagnostics uses molecular biology techniques to analyze biological markers in the genome, transcriptome, or proteome. The genome and transcriptome refer to the DNA sequence or RNA sequence that's present. The proteome is referring to the proteins that are produced. So when we say molecular, a lot of times people just think DNA. But molecular is all-encompassing of all types of molecules. And these molecules are the basic molecules that drive life. So we're talking about DNA, RNA, and proteins. Molecular biology or molecular diagnostics diagnose and monitor diseases, detect risks, and guide treatment decisions. Identifies specific genetic mutations, chromosomal abnormalities, or pathogen-related nucleic acids that are associated with various conditions, such as infectious diseases, cancer, and inherited disorders. It's highly sensitive and specific results, enabling personalized and targeted healthcare approaches. Molecular diagnostics has the widest applications across the clinical lab. Every area of clinical testing includes some type of molecular testing, and molecular testing is now replacing some diagnostic procedures. So some of our typical culture methods we no longer do in microbiology because now it's all done with molecular testing. Some other areas within the lab, we replaced those tests with molecular tests because we found that testing for the genetic material was not only more specific, but gave us a lot more information about what's going on inside the person. There is a major caution here. So just because a gene is present doesn't mean that it's expressed. So when we're talking about evaluating a person for the presence of a gene that has the ability to cause cancer. So if we're talking about like the breast cancer gene, BRCA. So just because a person has that gene, it doesn't mean that the gene is going to be expressed, meaning that it doesn't mean that the gene is actually going to be transcribed and translated into a functional protein. Even if it's expressed, so even if it is turned into a protein, it doesn't mean that that protein actually works to be functional to cause whatever disease we're looking at. So with the cancer example, it doesn't mean that it would actually go ahead and cause cancer. Even if it is functional, it doesn't mean that it is functional to the point of causing disease. So it may cause some disruptions in cellular function, but it may not lead to the full-blown disease. So we have to be careful when we look at genes. Just because a gene is there, we can't just say, yes, this person's going to have this disease or this going on with them, because we don't know if that gene is actually expressed. And this is why evaluating genetic sequences and DNA sequencing and everything surrounding genetics is so highly confidential. And there's a lot of ethical implications here, because if health insurance companies got a hold of certain sequences and they're like, oh, you were predisposed to all these diseases because you have the genes for them, that can increase your possibly health insurance rates or possibly life insurance you might be barred from getting. And there's a whole bunch of other things that go into this. But basically, we need to actually evaluate the genetics, see if the gene is present, see if the gene is functional, meaning that it was produced into a protein. And that's where the proteomics comes into play. And then also see if it did eventually lead to that disease state. Now, if we're using molecular diagnostics to see if a person has a particular pathogen, the presence of the pathogenic DNA may indicate that, yeah, the pathogen's there, or it may indicate that the pathogen was there previously. And if the pathogen goes away, sometimes that genetic material can't be detected anymore. So it's hard to determine if the pathogen's not there, that they even had the infection. So that's when other areas of the lab come into play, like immunology, where you may have those antibodies that are left behind after an exposure. So all of this is related and all of it's interconnected, but there's a lot of cautions here, and molecular diagnostics has to be carefully considered before results are released and things are said to patients and all that kind of stuff, because again, just because a gene is there doesn't mean that they have that disease, and just because the gene isn't there doesn't mean that they don't. So just a bit of caution. Specimen accession. So pre-analytical error is the consequence of erroneous or misleading results caused by events that occur prior to sample analysis. The condition of the specimen and, if necessary, the chain of custody is reviewed upon receipt in the laboratory. Chain of custody, that can come in a variety of different forms, like if we're doing molecular testing for paternity or a criminal case or forensics or that type of stuff. So then we would look at the chain of custody. No specimen is accepted without proper labeling and identification. If a specimen is unacceptable, the disposal or retention of the specimen is documented. All specimens are potentially infectious and should be handled with standard precautions. So this should all be pretty much a review of basic specimen handling. So gloves, that's part of the standard precautions. They protect nucleic acids, and they're absolutely required for handling of RNA. Transmission-based precautions, including respirators, are used with airborne or contact transmissible agents. Contact precautions are designed for direct patient care. Specimens of minimal cellular content are often analyzed. Cross-contamination must be avoided. Inspect blood samples for hemolysis. If cell lysis has occurred, DNA and RNA yield will be reduced. Why is that? Well, as soon as you burst open a cell, the DNA and RNA are free to go wherever. And there's a lot of enzymes inside of the bloodstream, inside of different fluids, that are designed to digest and break apart DNA and RNA. So if we have these enzymes, like RNase is present or DNase is present, then our DNA and RNA concentration is going to go down because it's all going to be digested away. Solid tissues are best analyzed from fresh or frozen tissues. The quality of nucleic acid from fixed tissue depends on the fixing process and the fixative used. So here are some anticoagulants for molecular tests. So So you can see which tests over here, the color of the top of the tube, and then what additive is inside of there. So our molecular studies are going to use these guys mostly. Immunology, virology studies, mostly green. And then your basic chemistry serum antibody studies would use the red top. Specimen holding and storage. For DNA, blood and bone marrow fluids, less than 1 day, you would put at 23 degrees Celsius. 3 days, 4 degrees Celsius. So 23 would be possibly around room temperature or a little bit colder. 4 would be in the refrigerator. White blood cells greater than a year would be -20 or -70. And that's in an intense freezer. Tissue, so 23 degrees, not recommended. Less than 1 day would be 4 degrees, greater than 2 weeks would be -20, greater than 2 years would be -70. Isolated DNA, less than 26 weeks, 2 to 25 degrees, so that's in the fridge or room temperature. 1 to 3 years, 4 degrees. Now when we isolate the DNA, typically we're going to isolate it in a sterile container, and that container is going to remain closed so that there aren't those DNA enzymes present to break down the DNA. Less than 7 years, we'd put it at -20 or -70. Blood and bone marrow fluids for RNA. So RNA is a little bit different because RNA is super unstable. As I mentioned, we will get into the difference of the structures of DNA versus RNA. RNA is typically single-stranded, while DNA is typically double-stranded. Double-stranded DNA is a little bit stronger and a little bit more stable. RNA, being single-stranded, is much more exposed and weak and able to be broken apart. So we have slightly different recommendations for RNA. Blood, bone marrow, or fluids, less than 2 hours, 23 degrees or 4 degrees. 5 days, 23 degrees. 7 days would be 4 degrees. 1 to 2 weeks, 70 degrees or -70. And white blood cells would be 2 to 4 weeks, -20. Greater than 6 months would be -70. Tissues, less than 2 hours would be 4 degrees. Snap frozen is also a possibility. -70 degrees centigrade would be greater than or equal to 2 years. Nitrogen vapor, -140 to -150 degrees. Isolated RNA, not recommended to keep it at room temperature. Less than 30 days, -20 in RNase-free water. -30, or less than 30 days, -70 in RNase-free water. And greater than 6 months, -70 degrees in ethanol. Laboratory preparation for RNA analysis. So the bench and equipment, you have to use a separate laboratory area that's designated as RNase-free. RNases are everywhere. They're contaminating in the environment. They're all over the place. So if we want to do any work with RNA, we would do that work conducted in a separate laboratory area. Disposables or reagents, certified RNase-free. Reactions, add RNase inhibitors. Federal regulations from the FDA require validation of the performance of clinical test methods and reagents in accurately detecting or measuring analytes prior to use in human testing. So, the criteria and definition are listed here. Detection limit or lower limit of detection, the least detectable presence of the analyte. Precision is the agreement between independent test results. So, are we getting the same value every time? Reproducibility is consistency of the test results produced from the same procedure. Analyte measurement range is the range at which a specimen may be measured directly without dilution or concentration. Reportable range is the range within which test results are considered to be valid, with or without dilution. Reference range, expected analyte frequency or levels from a population of individuals. Linearity, quantitative correlation between test results and actual amount of analyte. sometimes referred to accuracy. Test validation is performed on specimens of types that will be encountered in the routine use of the test. The number of specimens tested varies with the procedure and the availability of test material. Results from the new test methodology are compared to results from established procedures or correlated to clinical diagnosis. Commercially developed and FDA-approved molecular methods are verified by using the purchased reagent sets to test validation specimens in the laboratory. If the commercial test is modified, validation is required to show equal or superior performance of the modified procedure. So you can't just change up a procedure and expect to report out the results without answering to the validation of that new procedure. Once a procedure has been established, the method is documented in the laboratory according to CLSI guidelines. Proficiency testing refers to external specimens from a reference source supplied to independent laboratories. The College of American Pathologists and other organizations supply specimens for molecular analysis. If proficiency specimens are not commercially available, laboratories can exchange blind split specimens, or blinded specimens can be measured or documented by independent means, for example, chart review, and can be tested within the laboratory. Controls are samples of known type or amount that are treated like and run with patient specimens. With qualitative tests, a positive, negative, and in some cases, a sensitivity control are required. In quantitative methods, a high positive, low positive, and negative controls are included. For amplification procedures, amplification controls are required to avoid false negative results. Amplification is talking about when we do like polymerase chain reaction, or we make many copies of a particular region of DNA to be able to visually see that region of DNA. So if we make the copies, or let's say if we try to make the copies from a big segment of DNA and we run it out on a gel, which would separate those copies from the rest of the DNA, and we see that there's a band of those separated copies, then we know that the gene was present. If we try to make the copies and no copies were made, and then we run it out on a gel, which would have separated out those copies, and we don't see a band, then we know that there was nothing to copy. So when we use amplification controls, they're required to avoid those false negative results that may happen. Quantitative PCR methods that automatically analyze results require a standard curve or dilution series of a reference standard. So quantitative PCR, again, in this course, we're going to go through this stuff in so much detail, but basically it's going to, as the PCR, polymerase chain reaction, makes copies of the DNA, it's going to quantify how many copies are made so that you get a number behind the copies instead of just a visualization of yes, it's there, or no, it's not there. You get an actual quantifiable number. In methods requiring detection of a target-specific product or relative amounts of target, internal controls are run simultaneously, preferably in the same reaction mix as the test specimen. For example, housekeeping genes, centromere probes, amplification controls. Quality assurance is periodic review and documentation of test results are required. Molecular quantitative methods should have a defined dynamic range, sensitivity level, and accuracy. Assay levels that distinguish positive from negative results or cutoff values must also be well defined and verified at regular intervals. Manufacturers supply recommendations for routine maintenance. The laboratory maintains a schedule and instructions for all routine maintenance. There are limits of user-recommended repairs when service calls are indicated. Excuse me. Regular calibration or fitting an instrument or test system output with the actual concentration of a reference analyte is required for detection systems. Reagents. Instructions on the preparation of reagents and the quantities used in each assay are included in the written laboratory protocol. The sequences of primers and probes are documented. Primer binding sites and sizes of expected amplicons are documented. So as of right now, you may be unfamiliar with what a primer is, what a probe is, primer binding sites, amplicons. All of these terms may sound foreign to you. Throughout this course, we're going to be talking more about all of these topics. So for right now, you can kind of look up the definitions. Primers will help set the stage for a replication event. Probes are specific sequences of genetic material or sequences of nucleic acids that are designed to bind to other complementary specific sequences to identify sequences within large samples. Primer binding sites are the areas on the DNA where a primer would bind, and sizes of expected amplicons. Amplicons are what we call the copies of the DNA that have been made. So when I talked briefly about polymerase chain reaction, that's what those would be. So those would be documented. Analyte-specific reagents are probes, primers, antibodies, or other test components that detect a specific target. A target would be one of those sequences of DNA that we're looking for. Most ASRs used in the molecular laboratory are Class 1, and they're not subject to controls by the FDA. Class 2 include those used by blood banks to screen for infectious diseases, or those used in the diagnosis of certain contagious diseases such as tuberculosis. In vitro diagnostic reagents, in vivo and in vitro. You need to be able to distinguish between those two terms. So when we talk about in vivo is talking about within the living organism, so inside of an actual body. In vitro would mean outside of the body, which would mean that you would take part of the body outside and you would do something outside of the body. So if you draw blood from a patient and then analyze the blood, that would be considered in vitro diagnostics. IVD reagents include products used to collect, prepare, and examine specimens collected from patients. IVD reagents are classified according to performance risk and requirements for surveillance and verification from Class 1 lowest risk to Class 3 highest risk. In vitro analytical tests, reagents target a specific disease or condition. FDA will approve only the analytical validity of the test with no claims of clinical utility. Research use only. Those reagents are not intended for diagnostic use. While sometimes they can be used, the results of those could never be reported, or there's no validity to those results. Investigational use only. Reagents may be used on patients with proper institutional review and informed consent, for example, in clinical trials. Sometimes you're going to see these abbreviations, and they may come back on the qualifying exam later on in this course. So if you see these abbreviations, just know what they mean. This is another thing that you may see on your qualifying exam. The National Fire Protection Association has developed a series of warning labels. So what you may see is the yellow 1, the red 2, the blue 3, and the white W. And with those, you may be asked questions like, what does the red one mean? So you should be familiar with what these are. The red one is the fire hazard. The yellow one is reactivity, the white one is a specific hazard, and the blue one is a health hazard. Transport and storage. Secondary or reinforced containers are required for the transport and handling of dangerous chemicals, such as concentrated acids or phenol. Volatile and flammable reagents are stored in properly vented and explosion-proof cabinets or refrigeration units. Secondary containers are required for the transport and handling of biological specimens, for example, tissues, blood, DNA. Temperature controls, refrigeration packs, dry ice are required for stability of temperature-sensitive targets. Potentially infectious specimens are labeled appropriately. Nuclear Regulatory Commission requires that laboratories working within radioactive reagents maintain a radiation safety manual providing procedures for the safe handling of radioactive substances. Radioactive reagents are used and stored in designated areas. Test results in the form of electrophenograms, gel images, or autoradiograms should be of sufficiently high quality that results are unequivocal. Documentation of assay conditions, reagent lot numbers, and quality and quantity of the isolated DNA or RNA is required. In situ results, such as FISH, fluorescence in situ hybridization, which again we'll talk about later on in this course, are correlated with histological findings, stained sections of tissue morphology. Raw data are retained within the final report and in clinical interpretation of the test results. Reporting test results. So the test report contains patient information, laboratory and signee identification. The test results is accompanied by the method or commercial kit used, the locus, mutation, or organism tested, the analytical interpretation of the raw data, and the clinical interpretation of the analytical result. The potential for false positives or false negatives may also be included. The clinical significance of the test result and the references are also indicated on the report. Test reports and related data are protected health information and should be maintained by secure electronic or physical storage. And then you can review the summary slides.